Hello everyone, today we are going to discuss on a topic called restriction enzymes. Restriction enzyme. It is an enzyme that targets double-strand DNA and cuts only in the center or middle of DNA. Restriction enzyme is also called as restriction endonuclease, a protein produced by bacteria that cleaves DNA at specific site along the molecule. In the bacterial cell, restriction enzymes cleaves foreign DNA, thus eliminating infecting organisms. It is an enzyme that targets double-strand DNA molecule. They are specific in nature. They need support in the form of cofactors to perform their functions. Restriction enzymes are available many in number. The performance of restriction enzyme is called stack activity. Restriction enzymes were discovered and characterized in the late 1960s and early 1970s by molecular biologists Werner Erber, Hampton Smith, and Daniel Nathans. Here we can observe the types of restriction enzymes as type 1, type 2, and type 3, the cleavaging site, location of methylase, and the examples of type 1, type 2, and type 3 restriction enzymes. All restriction enzymes will have their own restriction sequence and own target site. For example, cut with SMA1, it produces a blunt end, and a cut with BAMH1, it produces a sticky end, and the cut with PST1, it produces a sticky end. As we have discussed, the restriction enzymes need a support in the form of cofactors to perform their functions. We can observe there are two types of cofactors. They are ATP-dependent restriction enzymes, metal-dependent restriction enzymes. And next, we can study about neoschizomers, isoschizomers, isocondomers. First, we'll discuss about neoschizomers. In neoschizomers, if any two restriction enzymes have same recognition site and different target site, then we call them as neoschizomers. For example, SMA1, XHO1. Now we'll discuss about isoschizomers. In isoschizomers, if any two restriction enzymes have same restriction enzyme and same target site, then we call them as isoschizomers. Examples are BDO1, SPH1. And then we'll talk about isocondomers. Any two restriction enzymes different in restriction enzyme and different in target site, but they produce similar ends. Then they are called as isocondomers. For example, NCO1, PAG1. And now we'll discuss about the properties of restriction enzyme. Restriction enzyme act on double-strand DNA internally at specific location, and the restriction enzymes are categorized as cofactors like ATP and metals. They can also produce two kinds of fragments, cohesive end fragment and blend end fragment. Restriction enzymes follow N+1 rule while making fragments. Restriction enzymes have their own recognition site and target site. Number of DNA sites decides the number of fragments. Now we'll discuss about the rules of nomenclature. The first restriction enzyme is Hind3, where the first letter in the restriction enzyme goes under the genus, the next two letters goes under the species, and the third letter goes under the strain, and the Roman number indicates the order of discovery. Each restriction enzyme is named after the bacterium for which it is isolated. The first capital letter indicates the genus, and the second capital letter and third capital letter indicates the species of the bacterium. Now we'll discuss about the types of restriction enzyme. There are three main types of restriction enzyme: type 1, type 2 and type 3. Type 1 restriction enzyme. They cut 1000 base pairs away from the target sequence. They have specific recognition site, but they cut 1000 base pairs away from the target site. Type 2 restriction enzyme. It cut at a particular location. Some do not get at exact position, but above or below one base pair of the target site. Type 3 restriction enzyme. They cut 23 to 25 base pairs away from the target site. Smaller the size of the base cutter, more the fragments formed. Now we'll discuss about the applications of restriction enzymes. Restriction enzymes digests the double-strand DNA so we can get our gene of interest. Restriction enzymes are widely used in restriction mapping studies. It is also used in hybridization studies and in sequence studies. And it is also used in polymerase chain reaction. Also, we'll talk about the drawbacks. Choosing a less base cutter is also a drawback. Choosing of restriction enzyme shouldn't influence our gene of interest. Large base cutter are used to cut unique fragments or to cut large space. Here we can observe the examples of restriction enzymes in a tabular form. Thank you.